This is going to be a discussion session called Choosing to be a Human Reference Standard. Very excited to have an amazing group of panelists, Misha, Angrist, Dorit Berlin, and Marc Salit. With a moderator from Radiolab, I'm very happy to say Robert Krulwich is here to lead the discussion. So please help me in welcoming the next panel.
Okay. I gather a lot of you are really doing this for real. So I have to, I'm going to try to advocate on your behalf and try to make you worry just a little bit, because I feel that's just necessary. But let me worry about ethics, worry about what you're going to know, what you're not going to know, why not, things like that. So to help me, I'm going to just introduce these folks. This is Dorit Berlin. She is principal, this is the longest title. She's principal investigator at the Coriell Institute for the National Institute of General Medical Sciences Human Genetic Cell Repository. Principal investigator mean like you're the boss? Or?
Well, that's not quite my title,
oh.
but you're close.
Wait. It says principal investigator, or is that like a?
Yeah, well, my principal investigator actually of the NIGMS repository, which is one of the biobanks at the Coriell Institute for Medical Research.
You said that so quickly.
Because I'm used to it.
Oh, okay.
Which is where the PGP blood samples come in, and we've got your cell lines, any of you who've submitted a blood sample for a cell line.
Do you have a picture of like where they are, their stuff?
We do.
Can I just so you know what's happened to that little bit of you, go ahead and show us your tanks, if that's not too gross.
Whoever can put the picture
Show this lady's
up.
tanks, as it were. Well, this is her name in the middle. That's a different slide. That's a different slide.
Okay, there you go. So on your left, you can see pictures of the insides of our liquid nitrogen tanks at the Coriell Institute. We're in Camden, New Jersey, and there's someone from cryogenics holding up, it's called a cane, and in the cane are stacked individual ampules of cell lines. So that's how we ship them, as individual ampules. And
wait, so does that mean like this thing here is Gladys? Or Gladys's tongue? Or?
Well, in there.
In there.
Yeah, Each of these is an individual.
Okay.
It's a glass vial that's been sealed.
What are the big vats?
And then those are the actual liquid nitrogen tanks. They're like, I don't know, pretty big around. And that's our tank room. We have over 100 of these liquid nitrogen tanks at Coriell.
And those are refrigerators really, right? Liquid nitrogen?
Well, freezers. It's really cold.
Freezers.
Yeah.
And what's your electricity bill every month?
Well, liquid nitrogen is what keeps it cold.
So it's not electricity.
It's gas.
All right, sorry. What's your liquid nitrogen bill every month? That's big.
I don't know, but we do get charged for it.
All right. And when we say biospecimens, I was just thinking before, so that's blood, right? People give you blood.
That's
people give you tissue.
Yep.
Did people give you like snot or poop or tears, or what else can you give you?
They could. Common things that we have at Coriell, we do have, like you said, blood, skin that we'll make skin cell lines out of, a lot of spit. For one of our personalized medicine
Like, is there a
project.
spit tank in there? Like, just spit we love? Or anything?
Well, the spit is a little different because that gets stored at room temperature, but there's lots of tubes of spit lying around. I don't tend to mess around with those.
You're showing a very unprofessional distaste for spit, I think.
It's good for research.
That's good. I'm glad. You have to be pro-spit on this.
Yeah, definitely pro-spit. Yeah, my daughter, they have all the company that makes the spit kits, DNA Genotech. They've got all these really great T-shirts, and like spit happens. looking
People
good.
walk around spit.
Spit happens, looking good in my genes. So every time I go to a scientific meeting and DNA Genotech is there, I try to get one of their T-shirts.
All right, enough of you. Marc Salit has worked extensively in measurement science, in chemistry and physics, with emphasis on very precise measurements in lab automation. I'm reading from his thing here. Algorithm development, measurement, uncertainty, traceability, and standards development. So what really this means is this is a person who doesn't like ever being sloppy, I'm figuring. So if you say to him inch or kilo or meter, for him this is something very specific. If I say cigarette to you, what happens?
I think we have a slide.
All right. You have like a Just So cigarette? Or
We have a Just So cigarette. So NIST has about 1,300.
That's a letter. That's not a cigarette. That's not a cigarette either.
That's a box of them.
Oh. It is?
It is. NIST has quite a portfolio of reference materials, some of which tend to be a little quirky. So we actually distribute reference objects that people can use either in calibration of a measurement or in validation of a meaurement.
When does this cigarette is too short or too long come up in everyday life?
Usually it's how flammable or non-flammable is your couch cushion. And so this is an object that burns it's been characterized to burn at a particular rate, and it is used in fire testing.
Really?
Yeah.
So you drop a burning cigarette on any number of couches you can choose, and while the couches may vary, your cigarette is always the same.
You can trust that.
Wow. And are firemen the people? I mean, just high school principals in smoking lounges? I'm trying to think, who's the customer?
I would imagine that there is some kind of underwriter's laboratory certification. I think there's a National Fire Protection Association, NFPA, that goes ahead and characterizes the very chairs we're sitting on for
You
health.
must get invited to very weird parties, as I'm thinking. All right, and then comes Misha, who is a professor at Duke University, where he thinks a lot about genetics and geneticists and how they should behave. And he thinks about this romantically, he thinks about it angrily, he thinks about it suspiciously, thinks about it cross-culturally, thinks about it descriptively, thinks about it critically, thinks about it in classrooms, in journals, in conferences. He's got his own body parts sitting in various biobanks. So if there's anybody you want to have on you have to have Misha on a panel like this. He comes with the chairs. I don't even say anything. I'll just open it with you, actually, instead. You just be quiet until I ask you this. You told me once that one of your most gratifying moments was you get a call from a professor at UC Davis who says he's working on you. What does that mean?
Yeah, actually, I was invited there for another unrelated reason. And you go to give a talk somewhere as an academic, and they hand you a list of people you're supposed to meet with. And on this list was a guy I'd never heard of, I think a cell biologist, and whatever, I got nothing else to do. Davis is a lovely place. And we're sitting outside the student union, and he gives me his story, and he tells me he's working actually on the CRISPR technology that George mentioned, this kind of newfangled approach to gene therapy, and that he needed inducible pluripotent stem cells, and mine were the only ones he could find, or the only ones that were available. I don't know. Maybe you guys were having a rough couple of weeks.
It was a sale. It was a Misha stem cell sale.
So he told me what he was doing, and this is what he was using my cells for, and it was gratifying. It was a sort of moment where I felt like this whole thing actually meant something and led somewhere.
I mean, like it's big. Like if you were a little bit of paint and I put you in the Mona Lisa, so you could be you could be part of a tree behind Mona Lisa's shoulder, then you're in the Mona Lisa. Like
that's right.
Is that the sort of pleasure? This is called I'm forever in some.
Yeah, I think we labor under this illusion. We go to the Red Cross and we give blood and we leave an hour or two later and we feel virtuous, and that's the end of it. I guess I feel like, boy, wouldn't it be great to know where this blood went, what became of it, what was actually being done with it.
So, that's the issue for everybody here, and for me, really, when I think about it, is the knowing part. Like, traditionally, you said this, genetics is something one does to someone else. So normally, if you're going to give a bit of yourself to a biobank or to a science project, what are generally the standard sort of way this normally operates? We take the bit of you, and then what do you get the right, what do they tell you you can know?
Nothing.
Nothing.
Typically. I sit on the Duke Institutional Review Board, and I review these protocols for biobanks and genetic research projects on human beings. And at least until recently, the SOP has been, you will get nothing and like it. You will get nothing because this is only research and the data are uncertain, and this is in your best interest to get nothing, because if you get nothing, then all of these fears are less likely to come to pass. Your identity being stolen and you getting cloned and probed by aliens and that sort of thing.
Well, before we get to probed by aliens, let's this means that not only will you know nothing about what the scientist is doing, but the scientist presumably will know nothing about you, other than your chemistry. Right? So it's a mutual nothing. How anonymous is anonymous really? Like, if I were to just take a little bit of skin and give it to a really smart detective, could that person, with knowing nothing but just analyzing, can it now work its way back to you?
Right. Well, if the detective had nothing but a piece of skin, they wouldn't be able to identify me unless they had something to match it to. And just in January, a paper was published in which they discovered a much easier way, well, not easier, but just with technology, it is easier way to match disparate sources of data to actually make that match between a person and that sample, even though the data isn't there with the sample saying that it belongs to that person.
So if I take a bit of your skin and I tell the detective that it's from America, or it's from America from a person born in 1969, or it's from America from a person born in 1969 living in Chicago. Like, how close do I have to get before the detective gets closer to you? You.
Yeah, well, there's a set of identifiers that we actually at Cornell, we don't put any of this information into our public database. So things like date of birth, social security number, name, phone number, address, other types of ID numbers. Any of these kinds of numbers are called personally identifying information, and they can use to track back to a person. So where it got really interesting for biological samples is that we used to kind of think of them as not being that identifiable, because how can you go from a blob of cells in a dish to knowing who they belong to? But really, our DNA within our cells is an identifier, essentially. So now that it's pretty trivially possible to sequence DNA or to sequence panels of variant markers, or to generate variant panels and know what those are, it's much easier to make that genetic match from a person to their sample, as long as there's somewhere else where that test might have been done.
As long as there's somewhere else. Oh, let me just give you a nervous example. I'm a young med student. I'm needing some textbook money. I go to the Fertility Institute. I give them one sperm sample every month, so that way I get beers and stuff. 30 years later, and I have spawned inadvertently 50 people, let's say. Now these 50 kids, they put their DNA things into some website, And the website, oh, look, X over there in New Hampshire is very, very, very similar to you in Vermont. So X in Vermont meet New Hampshire, and they talk about, and they realize that they're probably sired by the same former medical student. And let's say the number grows to 11. So there's 11 people who are now half brothers and sisters. And one day, Misha, how likely is it that one day these 11 people, having talked to their mothers, having got little bits of information about what their dad, then this poor man gets a phone call from 11 children he didn't know he had and has a heart attack?
Well, this is exactly what happened in 2005 when a teenager named Ryan Kramer did exactly this. Now, admittedly, he's a pretty precocious young man, but he took a Y chromosome test and he did some clever genealogical searching, and he had some other bits of information about his biological father, a surname, a donor number, and he tracked this guy down. You were mentioning this large SIB before. His mother, Wendy Kramer, went on to start something called the Donor Sibling Registry.
Right.
And I believe their biggest half SIB is well over 130 people, something like that.
130 people found out they were sired by this. Have they ever called up Dad? That would be like a Titanic of a telephone call. I would sink like a stone if I were on the other end of that.
Well, actually, one of the PGP-10, Kirk Maxey, was a sperm donor in the 70s and 80s, and subsequently found out that the samples he thought were mostly going for research. In fact, he probably has on the order of hundreds of biological children.
Wow.
And he has been open about this and would like to see, the way the industry is, the sperm banking industry is governed, change or be accountable to someone, which it really isn't at this point. Yeah, this happens.
Well,
this is reality.
So Marc, is this? If I were the dad, the putative dad, I would want to know: is this wrong? I mean, can you? If the phone call comes in and they say there are 100 of us, Daddy, like, can you? And I go to you because you're national standards. You say, well, actually, 93% of you. You can say that with a certitude of very high certainty, or
Very high certainty.
Very high certainty.
Yeah, I mean, it's essentially the human identity forensics, the kind of DNA forensics we see in the popular cultural icons like CSI.
You could learn it via 23andMe, and I wouldn't be surprised if it had happened already. There's a feature on there where they tell you, this is your likely second cousin or fourth cousin or whatever. But I mean, if it's a full sibling or a parent, you share half your DNA.
So can we then conclude that anonymity is promised, but anonymity is hardly assured? And as things get more and more, as people get more and more able through social media and whatever to meet each other and exchange information, your anonymity is very tentative at this point. Yes. What about if you're a researcher working in the lab? Now, here's what I'm thinking. I'm working on these cells. Oh, look, this isn't so good. I feel sorry for whoever that is, but I don't know whoever that is. I'll never meet whoever that is, and therefore I am under no moral constraint to warn, talk to, or contact whoever that is. If anonymity is being lost on the donor side, what is happening on the scholarship side? Any of you?
Well, I think there's still some denial at work, and this is probably a good time to give a plug for a side talk that's going to happen tomorrow evening in the Freedom Room, I think sometime around 7 maybe, where there's going to be a discussion of this set of guidelines developed by the American College of Medical Genetics. So you have genetic counselors and physicians ordering whole exome tests, that is sequencing of the 25,000 genes, and looking for a specific mutation that is causing disease in a child, let's say. But of course, you get all this other stuff, some of which may be useful or actionable. And so a couple of geneticists, Robert Green and Les Biesecker, and their colleagues developed guidelines for 57 genes that people doing these sorts of tests should return. And it has...
I'm not sure what you're saying. If I see something wrong in HTF101B, whom I don't know anything about, are these people in the Freedom Room going to say you now must, in certain circumstances, you should figure out a way to inform HTCBSB about this condition?
Well, the guidelines I'm talking about are for people you know who they are.
Oh.
The situation you're describing sort of goes back to what the paper that Dorit mentioned that came out in January, which says you can, what this guy Yaniv Ehrlich and his colleagues did at the Whitehead across town was to take so-called anonymous DNA samples and essentially do on a grander scale what Ryan Kramer did as a teenager, which is to take DNA sequence information and publicly available information on the internet, which now includes a lot of genealogy. Someone once said genealogy is the second most popular use of the internet. So it is possible now, I mean, he re-identified Craig Venter. He re-identified several families.
You mean he just got a bit of Craig Venter and they said, this is X. This could be one of 7 billion creatures on our planet. Who is X? And he went, that's Craig Venter?
Yeah, a few hours later.
Really? Oh, God. Well, then if a scientist, this is for you, if one of your people knows that you see something and like, oh, this doesn't look good, and anonymity is now so casual as to be sort of arguable for or against, isn't it rise to a moral place? Shouldn't this person? Let's suppose I'm looking at this and I say, oh, this person is 15 times more likely than normal to get a heart attack. But if he doesn't eat eggs all the time, he could reduce that. So someone should tell him not to eat eggs. And maybe it should be me or us. Do you have does your biobanks have an affirmative duty to go backwards up the chain and say, hey?
Right. Well, I would put out there that the situation for a biobank is a little bit different from the situation where an investigator is performing the research and has contact directly or indirectly with the subjects and is aware of all the analyses being done and gets the data and has a mechanism to get the data back to the subjects because, in fact, they had contact with them initially to do the experiment. Whereas with the biobank, we have no contact with the subject. We have very limited clinical data.
Because you're like the garage?
Well, we're a little better in the garage.
Those tanks are essentially black boxes.
Yeah,
but I mean, all of this has been decided by you guys just to make life easier for you. You could decide that when you take a sample from somebody, the person who gets the sample and stores the sample, i.e. you, the person who then studies the sample, i.e. you, should all make a deal. Like you should, oh, if we get alarmed by something, we have a moral duty to go up and tell someone, whether we're the garage or the person who learns it.
Well, actually, in the case of the PGP samples...
No, we're not yet, but I'm glad.
This is good for PGP. In the case of the PGP samples, because of the way the PGP participants are consented, you're consented for identification, you're consented for pretty much any use of your samples, and they can be given anywhere, they can be used for pretty much anything. And we have the PGP identifiers actually listed on the Coriell website for each sample. So you could Google your PGP identifier, find which sample is yours, and then click on the publications tab of your sample detail page and find what's been published using your sample that we're aware of. So
Can you even know, like, that Jerry at Duke is working on you, or do you just know?
Well, if they've published. Okay, so this is something that actually, it's difficult for us as the biobank to know exactly what's been done with the samples. Obviously, we know who orders them because we ship them out. But in terms of what's found with them, in terms of what's published with them, we ask people to report back to us, oh, when they purchase the samples, okay, let us know if any publications result from them so that we can be aware of that information and put that out for the community.
Yeah, but you guys are skirting my question. Oh, we publish. Well, we have some scholarly reason to publish. I'm just talking about my heart. I'm looking at this poor man or woman who has 15 times more likelihood to go, bleh, but I can help her if I could tell her, and you say, well, if it's published. I'm not asking you if it's published.
Yeah, but that's my point. My point is that people aren't giving us back information about what they published. They don't tell us we published this or that. We actually have to go out there and do literature searches ourselves to find what's been published. And whatever we can find, we pull in. But I'm sure that there's stuff that we're missing if they didn't cite it properly as coming from the NIGMS repository at the Coriell Institute. I mean, we even go and just do searches on Coriell because people don't cite their samples. Some do, so anyone who cites it properly, thank you, but a lot of people don't. And then we go out and do searches, but if we can't find it, then we can't say this research has been done. And obviously anything that's not published, well, they don't tell us that either, so.
Well, I mean, just going into the head of the researcher for a second, Marc. If I see something and I think, oh, well, here's a gene that looks like a bit of a mistake, or here's 5 genes that look like a bit of a mistake, or here's 50 genes. Well, if it's 5 or 1 or 50, that could be very different implications. And I guess what I would worry if I were going to call up Mr. X, I wonder, oh, have I got a correlation here, or have I got a cause or effect here? Do I know? Is one of the reasons why there is this opacity, is it because is that researchers are legitimately scared about certainty?
There's a history of technical artifacts, misidentification of a problem, but I would say that's not the dominant, that's not the source of where the tradition, and I would have to throw out that this tradition of anonymity for research subjects, I'm not sure where it came from, but we've been really careful to maintain anonymity as a space between clinical samples and research applications. There's been this firewall, if you will, between research and biomedical enterprise.
But is the best reason for that because you don't want to panic the person? You don't want to give them information that you yourself aren't sure about, or?
That's certainly a reason, but I might argue that the incentives to keep a person identifiable, as far as if I'm a researcher looking to advance my career, number one, I have HIPAA privacy to worry about, Health Insurance Portability and Accountability Act, which Dorit described some of these 18 identifiers. And so if I remove those as a researcher, if I remove those from the sample, then the sample is no longer considered a human subject. And so I'm free to do whatever I want with it. And the hoops I have to jump through are considerably fewer and less onerous.
Except that the universe you're examining is significantly impoverished by this. You don't know. Let's suppose you see someone with a lung cancer problem. Wouldn't you love to know whether this person has stopped smoking and if there's been a change, or when they started smoking? Wouldn't you like to know whether their parents were smokers? Wouldn't you like to know if they were addicts for smoking? Wouldn't you like to know, wouldn't you like to know, wouldn't you like to know? And you can't know any of that.
I can, but the IRB
Not if they're inhuman X's
Right.
on a sheet.
Yes, that's correct. You're preaching to the choir.
I don't want to do that. What about this? Won't this be solved by lawyers? Like, well, I mean, think about this. Like, I'm sitting there, and I have a 15 times more likelihood of getting blah, 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 and then I get blah, 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 and then somehow I hear that I have this problem because it was published. And now I go to my lawyer and I say, well, shouldn't they have told me? And don't they have a duty to tell? So the lawyer goes to court and says to a judge, this is shameful, and we now want this is now negligent. So they make it, and let's say it is. Now what happens?
So I think this is sort of the runaway train that we are on, and this is exactly what happened in the 1970s with amniocentesis. Amniocentesis was not routinely referred to pregnant women aged 35 or older until women started giving birth to babies with chromosomal problems and suing their gynecologists. And now amnio is offered without fail.
So then if I'm running an institute like you are, or at a school, right now I'm thinking, okay, let's give these people a consent decree. And I'll say, you know, of course, 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 let's please sign here. And you think, okay, now we're protected. We've told them that they're not entitled to anything ever, anywhere. Signed it. But now the lawyers arrive and say, actually, you have to say, we promise to, under the recent rulings of Gigmo versus Smigmo, we promise to inform you of this. So now the consent decrees actually begin to reflect the new law, and now that this consent decrees become more and more, I'm just making this up, so yes,
No. I mean, I think what you're asking for is for the researchers then to become medical practitioners.
Huh. Yeah.
Are they obligated to?
Yeah. Oh, so that's not good either.
It's different than what we're doing, that's for sure.
Huh.
I don't know. I mean, first of all, I would argue that that line is blurry and arbitrary. And I think it's less about trying to turn researchers into doctors than to just sort of have this framework, have this relationship reimagined to something more like a partnership than I am man in white coat in an ivory tower, and you don't really exist other than a data point in a spreadsheet or a vial of blood.
Well, let's run through a possible scenarios of how it might look like if you could just redesign it. One would be, who calls who? If we see something that makes us worried about the sample, is it the job of the science people to call the patient somehow or contact them? Or do you have to promise to call in every 2 years and say, what's up with me? So if you don't call, we don't ever have to talk to you. So who should go first? The researchers should contact you, or you should contact the researchers?
Well, I would like to have at least some mechanism for the researcher to contact the participant. There are some researchers who are very engaged. So, for example, you will have, say, physician scientists who work on very rare diseases. They know all the patients, they know all of their families, they come over for dinner or whatever. And you might say, well, is that really scalable? And I would say maybe not, but
how many people are in your batch? Like 10,000?
Oh, lots more than that.
Yeah, so she can't go to dinner with all of that.
But to your point, I think that this isn't the current practice, but I can imagine in the future something along the lines of a streamlined mechanism for reporting. I mean, if we had to go back, or whoever, like even if it wasn't on us as the biobank to report back, but then let's say it got pushed out to our customers who were actually performing the research on the samples and have the results, if they were then required to report back, there would need to be a pretty usable, reasonable mechanism so that they can actually get their research done and report the data instead of ending up spending all of their time reporting back data. Then they're not going to get any research done, and that doesn't help anybody.
Well, let's go from the patient's point of view, the donor's point of view. I promise you that you can see your genome every 2 years. We'll provide someone to help you understand what you're looking at. Or I promise to show you areas of possible concern, not the whole thing, but just things that you might want to know. Or I promise to show you even unevaluated information. I promise you may tell your spouse, your husband, your wife, your children, your grandchildren, or we would rather you not. Who talks to the insurance company? Are any of these, like, is there something, like how, should you edit this or not edit it? Should there be someone there to hold your hand when you look at this stuff?
Is the data, or is the answer right?
And is the answer right?
Yeah.
And so the current state of practice in clinical sequencing genomics is, that I know of, is you've got an academic research-oriented sequencing pipeline that happens in a research lab. When they have findings, they actually hand them over to a clinical lab for the clinical lab to reproduce under a regulatory oversight, essentially. So at that point, data can be reported back in a clinical context. That's clumsy at best.
Yeah.
I think
You keep calling some woman named Muriel or something, right? Who is this person that you say, oh, I'm just on the phone to, it's somebody, she works for George. But, oh.
Madeline.
Madeline. Yeah, who's Madeline?
Uriel.
Oh, I knew it was an M name.
Yeah, so Madeline Price Ball is probably here somewhere. I'm reluctant to speak for her, but she is, as far as I'm concerned,
She's probably fleeing from the room right now.
Yes, she's the PGP genome interpretation guru, and she has poured through hundreds of genomes and thousands of variants, trying to, along with a cast of thousands of volunteers, some of whom are my students actually, trying to look at every variant in every gene in every PGP genome and even some other genomes and figure out, is this likely to be of clinical importance?
And when you call her, is this like calling your PN or your physician's assistant at the doctor? Like, does she help you answer questions, or does she not do that?
Well, I don't know that I've called her. We communicate by email, but she would. I have every expectation that she would respond to any PGP participant, and now she's really going to hate me.
But I think you guys you'd want something like that.
Yes.
Like you want somebody to call about yourself. The other question I have is, like, when you use the word informed consent, when you sign one of these things, like, how informed must you be? Like, if you're going to ask people to sign a deal, then what do they have to know about genes? What do they have to know about cells?
Yeah.
Should you give them a test? Do these guys take a test?
They take a test, and they've all scored 100%.
Really? Mm-hmm. Well, see, now that's very weird, because that means you only get determined studiophiles. The medical community is not going to have the benefit of crazy people, people with no attention spans,
right?
People who are no
But,
will.
I mean, this is the thing: is
drunks and undisciplined people, which is a huge proportion of our genome.
Well, I might argue that there are some of those within the PGP.
Well, you have to score 100 on the test. So, all right, I'll give you a question. You've just been told you have a 15-fold above normal risk for getting Lou Gehrig's disease. Do you, A, do nothing, B, enroll in a program, C, shoot yourself? what is the right answer to that question?
Well, again, this is what my friend and colleague Michelle Meyer at Harvard Law School calls the heterogeneity problem, is we have different desires and different points of view about the way we react to information, what we want to do with that information. So presumably C means you are probably not the ideal candidate for the PGP.
Why? Shooting yourself might be totally rational response. If there are a few dead people at the end of every GET conference, you would say that that would be fully expected and they'd be carted off and, you know, put into a bus that is already waiting for them.
Because at least they got 100%.
Because they got 100%. Right, I mean, see, there's a kind of slippage that takes place here.
Mm-hmm.
Yeah.
But I think that you bring up a good point, which is that PGP is an incredibly well-educated, highly motivated audience, whereas someone who's going in for a test because their doctor told them to, not because they want to, they get a result. They're not trained in genetics. They don't necessarily know what it means. I mean, when you said that, my first question is, I mean, what's the, you know, how prevalent is Lou Gehrig's disease? You know, if it's 1 in 100 and my chances are 15-fold higher, then maybe I worry about it. But if it's 1 in 100 million and my chances are 15-fold higher, then it's still a very low probability. So I think it kind of comes back to education in the end, and we can't assume that people who are going in for any sort of a routine or non-routine genetic test have a particular level of knowledge about genetics or statistics or probability or what their results really mean. So it becomes very important to be really careful in how the results are reported back to the person. And in a way, almost the answer to your question isn't even in the consent forms that people sign. It's in education when they're in elementary school, middle school, and high school, so that they have the background with which to be able to understand these consent forms. I mean, I can tell you.
Well, apparently you guys don't trust the education system because you give a test of your own, which does mean then that it gets a little scarier to do. So people, like, you know, like frankly, in preparing for this panel, I look up him. When you look up the Google him, the one in the middle, he Marc. He's in there doing wheelies on some kind of a automobile, like literally. I mean, you look him up. It's Marc Salit, and it says like the first thing on Google is like there's some guy in a car running wildly through some motor place.
The same Marc Salit?
Same, yeah. Same. And then he's on some site. He likes a certain amount of, he's endorsing Keen sneakers because he washes them all the time and says they look really good. I'm thinking like, all right, so all of you are a peculiarly early adapter, let's try it and do it kind of people. You must have shy sisters, brothers, and cousins who would never be here, and therefore there's a skew in all this. And when you add a test to it, you're skewing it further. So you are all collectively abnormal, which for science is a problem, no? I mean,
it is a problem, but again, I think you're beginning from the premise that somehow 100,000 volunteers or somehow we believe collectively that the PGP is for everyone, and it's not.
Well, on the other hand, the PGP may not be, but this business of collecting samples, first of all, how rich, like if I'm a top 10, a Big 10 school, and I got my football team, I got my basketball team, and now I got my biobank, which makes me the most money?
It's not the
How good is your team?
Not how good is my team? I got a middling team. I got a middling basketball team. We get through halfway through March and we're out. But my biobank is growing, and I can charge. I don't know what you guys charge, but isn't this one of these things that if you're on Wall Street, you would bet on? Like, there's going to be more of this. You charge fees. The fees go to the school. That's got to be. Presidents should take note. No?
Well, our biobanks, a lot of the big ones at Cornell are funded through various institutes at the NIH.
Yeah.
And as
so you're a socialist. I'm talking about real American presidents of college.
Yeah, this is important because they fund us to process these specimens and QC them and store them and distribute them. And then, so the fee that's charged, it actually doesn't go to Coriell, it goes back to the owner of the biobank or whoever we have the contract with, whether it's the NIH or whoever, to offset the costs of running the service that we provide. And I'll tell you, they have to fund us because the amount that the samples bring in is less than the amount that it costs to provide the service.
Oh, so this isn't a high margin business yet.
Well, we also intentionally charge less than what it costs to provide the service, which was a decision made on the part of the institutes funding the biobank, so that they're actually funding the resource so that people can purchase them at an economical cost and really accelerate research, because not only do they not have to recruit the subjects, do the consent, get the specimen, grow the cell line, get all the data, we do all that for them, but then we also are able, because of the way it's set up with the funder, with our sponsors from the NIH, to offer it to them at a low cost. So it's basically a way of helping to subsidize research.
So that I'm wrong to think that this is like a big...
Not necessarily. I think it's an open question, and I think various universities and private groups are trying to figure out how to monetize human tissue and biological information, and time will tell. I expect some of them will succeed.
What about recruitment where you don't really, where it's an opt-out? So apparently at Vanderbilt, the plan there is if you go to Vanderbilt and go to the doctor on the campus, they just take your blood or take your tissue, whatever, and you're in unless you say, I'm not. Now, is that right?
I'm not particularly enthusiastic about that model. I can tell you it's been great for their recruitment numbers. So I don't know.
I mean, the tanks in Tennessee are huge.
They are.
Yeah. What about the U.S. Army or the U.S. Navy or the Marines? Like, if you're in the service and they take your blood, right, at some point, are you now biobanked somewhere if you're a veteran?
Well, George actually talked about this morning. The Million Veteran program actually aims to sequence the genomes of 1 million veterans. And as he pointed out, despite them having surveyed the veterans, the overwhelming majority said, yes, we want our data back.
We want our data back?
Yes. The VA said, well, we're not going to be doing this in a clinically certified lab, and so therefore that prevents us. So, sorry.
I mean, you went to Fallujah, put yourself at risk, we took your blood, and you can't have it back.
Well, the Million Veterans Project is by volunteering.
Right. Oh.
They volunteer for the Million Veterans Project.
Oh, okay.
So they're consenting in. They do, the Army or the Armed Forces do take blood samples for human ID and archive those on paper cards, which then are extracted and used in identification.
By the way, how much time do we have? It's now 11:45. When are we supposed to be done? 13 minutes? Okay, good. Well, shouldn't somebody in Congress say, well, wait a second here, like this is all kind of happening without really, talk about informed consent. This is sort of seems to be happening automatically, and it's a bit of a yank rather than a survey. Or no, am I exaggerating this?
You're talking about military specifically?
Military specifically.
The military is kind of a different beast. So the military is not included in the Genetic Information Non-Discrimination Act. And until about 2007 was actually the most prolific genetic discriminator.
What does that mean, prolific discriminator?
Well, so take this soldier who goes to Fallujah and loses a limb and comes home and gets care at the VA and a prosthetic or whatever. Maybe he has to wait a long time to get that, but that's a separate question. But learns that he has retinitis pigmentosa, a congenital disease that manifested late. And until this expose in the LA Times in 2007, the Army would regularly say, that's a pre-existing condition, that's not our responsibility. And so it was only when they were sort of shamed into covering genetic disease that changed.
So that's interesting. So as John said in the session before, sometimes it's lawyers, but sometimes it's reporters who make things. All right. Let me just propose a few things for you as we conclude. Let's just know, if we were just to invent from scratch a protocol, I'm going to just give you a contract to sign. I'm just wondering whether you think it's a good one. Dear participant, thank you for donating your sample. We have given it to the following laboratories for the following studies, and we will regularly update that list. We are examining your sample for cancer, heart disease, ALS, and we'll update that too. Thank you for participating. Goodbye. So what you're learning here is that we've taken a bit of you. It's going to these places. You can at least know who's looking at you. Any problem with that?
That doesn't, I'm not sure what you're getting at. That doesn't sound like a consent.
Oh, yeah. What is that? That's called a...
Yeah.
What is that? That is just something
That's a Christmas card. And in fact, I was assigned a biobanking protocol on the Duke IRB and agitated for exactly that sort of communique with participants. You know, once a year, here's what's going on with your sample. And thanks. And I was told that that was impossible. It was logistically impossible. It was a HIPAA violation. And you know, this biobank was just going to be a disinterested third party and couldn't possibly keep track of all of this stuff.
And that was resolved against you in that case? Clear by everybody? Or,
Correct. I believe the final vote was 18-1 against.
How about, thank you for donating, this is another Christmas card. Okay. Thank you for donating your sample. If you like, you can call this number and ask us who's looking at me. So no, it's your option. And we will tell you and update it. So it's the same thing, but one step. Now you have to ask. Is that better for your nameless university?
Well, you know, I don't know if it's better logistically, but again, I think that's a very skeletal version of what the PGP already is, which is, here is your data. You can log on and look at it 24/7. You know where we live. You know how to get a hold of us. So I think that is those examples are really just the tiniest of baby steps. And my hope is that the PGP and the other initiatives that George mentioned are at least a little further down the road.
Well, here, a little further would be, thank you for donating your sample. You're doing a wonderful thing for science and for the world. We can't talk right about what's happening to your samples at this time, but if something important turns up, we hope to let you know. Please trust us. George Church is our leader. He's big, he's cuddly. He does fall asleep rather suddenly, but he's always thinking of you. So that's a model where you get a kind of a Santa Claus, sort of. He even kind of looks a little Santa
Why
Claus.
bother?
So is there any like we're thinking of you? Is that like because George would be the first person and has said over and over again, do not trust me. He always says that. So I'm a little worried that this has the potential to be we care. And people will.
Well, there's no point to the third letter, right? I mean, it's just like almost insulting, really.
Yeah, except you know there are people who will sell services around trust and around, that's why you have food companies that will put people on what are those, like, you know, recipe companies. I wish I had better, see, in my genome they would have called proper nouns leave you just when you need them problem. But there are foodstuffs, you have to help me here, where you see a lady on the bottle and she's going to take care of you and you trust her, even though she's not a real person.
Oh, like Aunt Jemima.
Thank you. Aunt Jemima. So yeah, so George could be the Aunt Jemima of genetics. I could just have this. It is silly, but it's not completely silly.
I understand what you're saying, and at this meeting last year, I remember witnessing George being greeted by a man in a wheelchair who turned to George and said, "You're my last hope." And you know, I was sort of both moved and stricken by that, because you could do a lot worse than George, God knows, but I mean, where are we? What has it come to that this research study is someone's last shot at trying to find an answer as to what's going on with them?
Well, here's my last question, and then if there's any time, I'll ask you guys to ask us stuff. But the last question is what, given the protocol that they have here, what dangerous new thing would you like to add to it? What should it do more of?
Oh, I can answer. I would love to see what people do with their data. I mean, so we give you oodles and gobs of data, and you can look at it 24/7. Well, what do you do with it? Do you change your life? Do you decide that you want to exercise more or eat differently or sleep differently? Because that's ultimately all that matters in the end. You know, research is nice for researchers, but in the end it comes back to the person and how does it have a positive impact on your lives.
What if you got a very disappointing answer? Like, I don't know. Or there was nothing here I understood. Or I use it as bragging rights. It's like having a Mercedes, you know. Oh, I have a Mercedes, but now I also have a personal genome.
Well, I think, yeah, that's something really important for scientists to look at, right? Because if it ends up not being used to actually improve life for people, then we're doing something wrong, and we would need to fix that or do work towards fixing it.
I'm with Dorit on getting back sort of a meta study of what's the impact of having your genome. I'm still really interested in the idea that the PGP could be used to bridge that gap between medical practice and research. I'm still worried about the high school student who downloads a genome and develops a new algorithm, not really vetted, not really tested, but publishes something, maybe self-publishes in an open journal that's not particularly critically reviewed. And now we've sort of spilled out this possible reporting back of bad news that may in fact be fallacious. So
you have problems with chat groups or listservs. So if I'm signed up with a group of people who are like this one, and we just go offline and we meet in cafes. We have Thursday genome nights.
Salons.
So yes, salons. So we can tell each other all kinds of crazy fibs and get into quiet panics and noisier panics. Is that what you're worried about?
I'm not worried about I'm worried about people taking actions based upon wrong answers.
So is your answer to your question, what would you improve or add to the protocol, is called don't?
No, I think the chances, I think this is one we'll figure out by crowdsourcing it, frankly. I think that the more this happens, if we get to 100,000 personal genomes, new norms will arise for how to handle this information. Right now, I think we're immature. And we're in the early adopter lunatic fringe, you know, tail of the distribution. Where are we? Hey.
Well, you're with your
these are my people, right?
We have each other.
We have each other. And so I don't think we have the cultural norms or the social norms or the practical norms to deal with this yet. So we have to push forward and make that happen.
Misha.
You know, I think if we really pay attention, that a lot of this stuff has already penetrated the culture. So patients like me has, I don't know, 200,000 participants. And these are people who have generally, if they've not explicitly foregone privacy, they're not paying very much attention to it, and I'm sure Ben Haywood will talk about that. And they have Lou Gehrig's disease and depression and multiple sclerosis and these horrible conditions in many cases that have led to a lot of suffering. And they are looking to each other not only for solace and commiseration, but actual ideas as to how do we go about changing this. And the idea that it's somehow stigmatizing for them to do this it doesn't even enter into the equation. So I'll tell you a story. So I assigned my students a paper about re-identification. It was not the one in science, but it was simply talking about this idea, and Jim Watson not wanting to know his ApoE Alzheimer's risk, et cetera. Unusually, I had not read the paper before I assigned it. And so I finally sat down to read it, and there was this discussion of one of the PGP participants openly sharing his past history with depression and anxiety and SSRIs. And there was a quote from him, and I looked at it and I said, oh, that's me. And what I was struck by was not embarrassment or, oh my God, my students will learn this about me. I was struck by the fact that this was a big deal at all, that this was somehow worthy of attention because I had included this bit of medical information about myself in the context of DNA, that somehow DNA makes it all scary and dangerous and
or more like you're a young med student and you're working on a corpse, and they work hard to make sure that you just see parts of it. It's an it, it's an it, it's an it, and then you see the pink fingernail polish.
yes.
And you think, oh, that was a person. Is that what you're saying? That you in that moment you came out of the data and you were fleshy, and troubled.
But I think it's fine because we are people, and you know, there's a scene in Rebecca Skloot's book about Henrietta Lacks that does exactly that, where George Guy's assistant sees her in the morgue, sees her painted toenails, and says, "Oh, geez, she's a person." And when I saw this little quote from my PGP profile in this paper, you know, it just seemed so gratuitous and such a sort of an attempt to alarm that, you know, this is the can of worms that we are opening. And I just thought, well, if we're going to continue to look at this sort of information with this stigma attached to it, then it becomes self-fulfilling.
So then we're going to conclude. You're going to conclude saying, "What really? This is a study by humans of humans, and though the hearts may not be, well, they are beating. So you should listen for the beating heart in this information. It's not just facts. It's got a pulse." That's not a bad place to stop. We have 5 minutes or so for questions and answers. And says, are you waving wildly in the back? You are indeed. Okay, well, can you just shout out whatever it is you want to know?
If the microphone works, yes.
Oh.
I'm a privacy researcher from Switzerland.
Yes.
And we have in Germany and in the European Union strict privacy regulations. But as Misha Angrist said, there seems to be a firewall between the area of research and the area of clinical practice. Privacy regulations only apply to clinical practice, but not to research. So my question to the panel is that a hindrance to what you're talking about wanting to do, or is there maybe potential in the fact that research is exempt from privacy regulations?
Research is not exempt from privacy regulations.
Not in this country.
Not in America.
We have very strong, I mean, every time someone wants to do research with a human subject, they have to submit an application to their institutional review board. It has to get reviewed. They have to get approval. All the consent forms have to be reviewed. In fact, I did a project a few years ago where it was developing an educational teaching tool for middle and high school students. I had to submit the protocol to the IRB just to be able to go into the school and have the students view the module and answer a survey.
But aren't you chilled? Like in the Canadian session this morning, the takeaway there was Canadians seem to be immodestly interested in themselves. Which, who knew? So maybe the privacy issues are maybe we're overemphasizing them.
Well, again, this is force of habit, because in all of these consent forms I see every month, there's extensive stuff about privacy, and there are federal privacy regulations, and there's GINA, et cetera, and then there are all the exceptions and caveats. Well, unless the FDA wants to review it, or NIH, or the sponsor, or the drug company, or the IRB, and then all bets are off.
And also consent forms change over time.
Right.
So consent forms, you know, one could perhaps make the argument that maybe in today's world of Facebook and Twitter and Google and all the social media we have, that a lot of our consent forms might be outdated because we have a population now that really wants to put their information out there. You know, I can go onto Facebook and people reveal all sorts of things about themselves, way more revealing potentially than what a whole genetic sequence would reveal. And you know, so in a way, our consent forms, we're so worried about protecting the human subject and thinking that, oh, researchers, you know, people don't want to be identified. They want everything to be private, when in fact, some people want to be identified. They want to be public. They want their data to be out there, and they're comfortable with it.
Of course, in Germany, they have laws which have the right to forget. Like apparently, if you do some embarrassingly silly thing when you're 19 and it's all over the internet and you're German, you can just go to somebody and say, forget about that. And everybody has to, including news organizations. So you're from a whole different world, you aisle number one. Last question.
I'd just like to make a point about the changing norms around this. There was a paper that came out in Biopreservation and Biobanking, which is a journal, recently from Biobank Ireland. And out of 400 members of a biobank there that were collected, the samples were collected in routine care, only four, indicated that they wanted each novel study using their sample to recontact them for permission. So 359 members, and these are people whose samples were taken during routine clinical practice, these aren't oddballs like us, thought it was completely normal for their samples to be reused for whatever research purposes.
Huh.
But again, I think that's the disconnect between what people say they want. I mean, you know, the military said, we know you told us you want your data back because we specifically asked you, but we're not going to do it.
All right, next up, I believe, is Jack Hitt, who is always fabulous. So you don't want to leave. Sure?
Important. My name is James Shirley, and I'm PGP 10. And earlier it was said that PGP 10 was the person who had the many children from sperm donation. So if you...
No, I said one of the PGP 10.
If you're looking for that, and you're searching PGP 10, you're not going to find that person, you're going to find me.
We should just tell you that PGP-10 is otherwise known as Genghis Khan, great-grandpa of 1/10 of all of Asians. All right, well, thank you very, very, very much. And thank you.